Animal testing is one of those things that seems necessary until you start asking the harder questions. What are the regulations actually based on? Are there any viable alternatives? And how much of this is science versus just tradition? Let's unpack the ethics, the system, and the future of biomedical testing. Why animal testing still exists. Most medical devices and pharmaceuticals must go through preclinical testing before human trials. This typically includes in vitro testing, or cells in a dish, and then in vivo testing in living organisms, usually animals, and it's rodents in about 95% of cases. So, why animals? Many biological interactions, like drug metabolism, immune responses, surgical complications, etc., cannot be fully captured outside of a living system. Regulatory bodies like the FDA, Health Canada, and EMA often require animal trials to prove safety and efficacy before approving human trials. These studies follow good laboratory practice standards and are reviewed by institutional animal care and use committees. One of the better reasons for animal testing, I think, is that it's much quicker to study the effects of therapies and diseases over the course of lives and through generations of animals with shorter lifespans than humans. This means that if we can catch some of these long-term ailments or side effects earlier in animals, we can prevent many times more humans and animals from being afflicted by these same diseases or side effects through the course of their lives. In either case, if a drug is approved that causes issues, how many more people are going to be affected by that than the animals in study? And if a drug never makes it to market because it can't be approved, how many people who would have had that therapy are going to suffer as a result? As I alluded to earlier, there are also positive downstream effects in veterinary medicine. The results of drug testing on animals are often used to inform treatments on animals, not just humans. There are, of course, also practical and legal considerations here. The scientific method demands an isolation of variables. Sometimes this means controlling light, diet, exercise, and interactions with other organisms. Sometimes it means inducing deadly diseases, neurodegeneration, cancer, oftentimes an implant or a resection of some tissue. The point is, many of these would run way against the Nuremberg Code, which was an agreement from 1947, which was adopted internationally, that outlined some of these ethical considerations for human testing. So this is the paradox, right? Animal testing is considered the ethical gatekeeper before human use, yet it directly compromises the well-being of other sentient beings. The foundation of modern animal research ethics comes from 1959, and it's known as the 3 R's. Number 1, replace animals with non-animal models wherever possible. Number 2, reduce the number of animals used. And number 3, refine the procedures to minimize pain and distress. Many institutions now have in-house veterinary staff, enrichment protocols, and humane endpoints. Humane, in this context, still often means terminal studies. I work in surgical device design, so all of our animal trials end in sacrifice. The recovery from these surgeries would be excruciating. Okay, so we have some understanding of why animals are used and some of the underlying ethical considerations regarding their treatment. But we still have to do our verification and validation here. And the first question that comes to mind for me is, do animals actually predict human outcomes? And here's the uncomfortable truth. They usually don't. So a 2007 review in BMJ found that many animal studies do not accurately translate to human clinical results. And according to the NIH, even with drugs, 92% of therapies that pass in animal studies fail in human trials. So why do we still use them? It's partly because our regulators still trust the precedent, and partly because our alternatives are not yet mature enough to fully replicate the complexity of human biological systems. But wait, there are alternatives, so let's talk about them. Organs on chips can simulate organ-level responses using microfluidic devices lined with human cells. 3D bioprinted tissues can replicate skin, liver, and even tumor environments for drug testing. AI and in silico modeling can simulate pharmacokinetics, which is how drugs move through the body, and predict adverse effects. And improvements in ex vivo perfusion mean that we can test on donated human tissues or organs post-mortem. Some of these are actually already FDA accepted in limited use cases, like the Tox-21 program for chemical toxicity screening. So what can we do differently, whether you're an engineer or not? I think in all cases, we can't just follow protocols. We have to be committed to building better ones. So here are my 3 suggestions for how to think about this differently. Number 1, design for data efficiency. The more you can isolate key variables in benchtop or chip-level testing, the fewer animals you'll need to train. Number 2, plan your failure points early. If your design is likely to fail, make it fail before in vivo testing. And number 3, advocate with your team. Sometimes engineers don't get a say in protocol design, so speak up. All of your design decisions are ethical decisions. Test for system fidelity, not tradition. A system that defaults to animal use without asking whether it's the best model is due for a redesign. Use proxy systems to isolate complexity. Human cell cultures and chips let you test specific variables without the whole animal confounders. Learn regulatory fluency. Knowing how IACUC, FDA, and EMA approvals work lets you propose validated alternatives confidently. Push for open data sharing. Redundant animal studies happen because researchers don't share failures. Design platforms that do. Reframe progress to include ethical cost. Engineering isn't just what you build, it's how responsibly you build it. So, can biomedical innovation be cruelty-free? Not yet. But it can be cruelty-reduced. And with the right incentives, like policy changes, funding shifts, cultural reframings, it can maybe be cruelty-replaced. It's not going to be a binary switch. It's going to be a transition, but we are all responsible for accelerating it. So I'm so curious to hear, do you think animal testing should ever be allowed? Are there ways to reduce the cruelty or the suffering involved? Or how would you reframe the policy to support animal rights? And if you work or study in a different field, I'd love to know what assumptions are made in your field about animal rights and the ethics surrounding animal testing. Thanks for watching, and take care.